Hello and welcome to week 1 of the third term of Anatomy and Physiology, where this week we'll be looking at chapter 20 entitled The Lymphatic System. Of all the systems of the body, the one that's, I would say, least familiar with most of you is the lymphatic system. And you'll notice in our textbook, it represents just a small section of material to learn in the course, but it brilliantly bridges the gap between our study of blood and blood vessels from chapter 19 and the more formal study of the immune system in chapter 21, the topic of next week's materials. And although I've just inferred that it might just be a connector between two chapters, don't get me wrong, the lymphatic system is very important in our fight against pathogens and in terms of body fluid, homeostasis. It's an important chapter to understand, so let's go ahead and get started. In this chapter, we'll first take a few minutes to reconsider the process of bulk flow, and thereafter, we'll discuss the 4 major functions of the lymphatic system. We'll then look at the 5 major components of the lymphatic system: our lymphatic fluid, the vessels that transport this fluid, the cells found associated with the lymphatic system, the tissues that are involved, and finally, the organs of the lymphatic system, both primary organs and secondary organs. So these will be the topics of this particular lecture. Now, before we begin talking about the components of the lymphatic system, I said I wanted to take a moment to review bulk flow. So we're going to look at some terminology and just general information about what we covered at the very end of chapter 19. Bulk flow is the physiological process. It's a passive process by which fluid and dissolved substances filter freely through the capillary walls, so our blood capillaries, into the surrounding tissues, creating interstitial fluid. They're bathing tissues and the cells with fluid, with oxygen, with nutrients, with hormones, picking up things as well. And that movement is driven by pressure differences between blood in the capillaries and the interstitial fluid within the surrounding tissues or surrounding those tissues. And we'll cover that in our next slide. There are 2 terms we need to consider here. First, filtration, and then reabsorption. These are the mechanisms that aid in the process of bulk flow. And when we talk about capillary beds, at the arterial end of a capillary bed, we have filtration, or the pressure-driven movement of a fluid and solutes from blood capillaries into the interstitial spaces. Again, the fluid carries nutrients, oxygen, other small molecules. They're pushed out of the capillary bed so they can provide support to the tissues and the cells making up those tissues. That is filtration. But then we have reabsorption. And as blood moves through the capillary bed toward the venous end of the capillary bed, we've pushed fluids out. Now we need to pull things back. We need to reabsorb things back. And so we'll see the return of fluids. We're going to see the return of local waste products from tissues that move back into circulation as well. So we have filtration and reabsorption as it pertains to these capillary beds when we consider the process of bulk flow. Now, in the last slide, I generically described the processes of filtration and reabsorption. But recall from chapter 19, we have specifically named pressures associated with bulk flow across the capillary bed. And the effect of these pressures depends on what part of the capillary we're considering. That is the arterial end or the venous end. And for the first part of the capillary bed, the part we call the arterial end, we're going to see hydrostatic pressure of the capillary. So we're looking here at this capillary hydrostatic pressure, abbreviated HPC, which is generated by ventricular contraction of the heart. This pressure, which I describe as the pressure against a wall, ultimately causes fluids, the nutrients, dissolved gases to be forced out of the capillary bed into that interstitial space, providing support for local cells and tissues. So when we talk about hydrostatic pressure, think about pressure against a wall. When we consider specifically capillary hydrostatic pressure, we're talking about the pressure of blood against the inner wall pushing outward. Now, in contrast, we also have osmotic pressure in the capillary. So now we're going to look at this colloid osmotic pressure. We're going to look at capillary colloid osmotic pressure, which is abbreviated OPC. As hydrostatic pressure decreases as we're moving through the capillary bed, the osmotic pressure is going to become higher, causing fluid to be pulled back into the capillaries from the interstitial space. Now, what makes that happen? Remember from chapter 19, we said proteins stayed in blood. Water is attracted to proteins. So as we see blood with a number of proteins moving through these capillary beds. We've pushed fluid out, protein has stayed within the capillary beds. Suddenly we're going to see the attraction of fluid to these proteins, and we will see the movement of fluid back into the capillaries at the venous end, or toward the venous end of the capillary beds. So those are our two major pressures, the capillary hydrostatic pressure and the capillary colloid osmotic pressure. Now, what about these two? Interstitial fluid hydrostatic pressure and interstitial fluid colloid osmotic pressure. When we're considering those two, we are looking at, when we consider the interstitial fluid hydrostatic pressure, we're looking at the pressure of interstitial fluid being pressed against the capillary walls from the tissue space. And then finally, when we consider the interstitial fluid colloid osmotic pressure, now we shouldn't have many proteins out in the interstitial space in a healthy individual, but on occasion, because of inflammation, and we see some leaky capillaries, we might see some proteins escape out into the interstitial space. And at that point, we would actually see fluid being drawn out of the capillary beds or having a difficult time moving back into the capillary beds. So we do see more fluid buildup that way. So these are our two terms, hydrostatic pressure, colloid osmotic pressure. What I want you to think of generally when we talk about hydrostatic pressure, it's a pressure against a wall. When we talk about colloid osmotic pressure, I want you to be thinking of proteins and the attraction fluids have to proteins. Now, I'm not going to test you on our net filtration pressure. However, as we review these concepts, I want you to understand what forces contribute to the inward and outward flow of fluids between capillaries and the interstitial space. And with all this in mind, remember from last term where I said, on average, we push out about 20 liters of fluid per day at all the capillary beds throughout the body. So we take all the capillary beds and we add up how much fluid we're pushing out due to the capillary hydrostatic pressure. We push out about 20 liters, more toward that arterial end. And then we're going to pull back roughly 17 to 18 liters of fluid as a result of our colloid osmotic pressure in the capillaries. What's left is about 2 to 3 liters of fluid in the interstitial space that must be drained, and that's going to be courtesy of the lymphatic system. So here we're connecting chapter 19 with chapter 20 with our lymphatic system. To introduce the lymphatic system, I want to go ahead and talk about the 5 major functions of our lymphatic system. First of all, the lymphatic system conducts interstitial fluid from tissue spaces back into the blood. And recall from last chapter, we bathe our tissues and the cells, the blood capillary levels with fluids, with ions, oxygen, we deliver nutrients, we see hormones delivered, and that all ends up in the interstitial space. And so we had that 20 liter pushed out, 17 to 18 liters brought back in because of those colloid forces, leaving 2 to 3 liters of fluid behind. And so, as I mentioned in the last slide, it's the lymphatic system that helps maintain fluid balance in the body by collecting excess interstitial fluid. We have lymphatic capillaries that are intimately associated with our blood capillaries that will ultimately help return fluid back to the circulatory system with the help of not just our lymphatic capillaries, but then we go on to create lymphatic vessels, to lymphatic ducts, etc. And maintaining fluid balance ensures that we don't accumulate fluid in the tissues or otherwise experience edema. So that's function 1. Function 2, we conduct leaked proteins from tissue spaces back to the blood. Now, in a healthy individual, we shouldn't be pushing proteins across the capillary beds. Proteins are typically too big to move across the capillary beds from blood to the interstitial space, but on occasion that happens. And on some occasion, such as under high pressure during inflammation, we might see small plasma proteins that leak out of those capillaries into the interstitial space. And although most fluids can be reabsorbed at the venous end of a capillary bed, not all the leaked proteins can be reabsorbed due to their size. And as a result, the lymphatic system ensures that any proteins that have inadvertently leaked from circulation to the interstitial space can be pulled back and can gradually make its way back to circulation. And again, this is courtesy of the lymphatic system. So we are going to see fluids pulled back in. We're going to see leaked proteins pulled back into blood. Next, dietary lipids and lipid-soluble vitamins like A, D, E, and K are going to be absorbed within the gastrointestinal tract courtesy of some specialized lymphatic capillaries called lacteals. These lacteals are found in the microvilli of the small intestine, and unlike other dietary substances, which are just going to be absorbed directly into blood, our lacteals are going to be responsible for absorbing those fats and fat-soluble vitamins. And we will find that they actually enter blood and then can become filtered along the way. Fourth, we see the lymphatic system aid in filtration and phagocytizing foreign materials. Now, I'm skipping ahead of today's lecture a little as I say our lymphatic system is responsible for the filtration of foreign materials in our lymph fluid and in the phagocytosis of foreign materials. So I'll say this for now. We're going to rely on our lymph nodes, and this is where I'm skipping ahead. We're going to rely on lymph nodes scattered throughout the tissues of our body to filter lymphatic fluid, and with the help of certain immune cells like macrophages that reside in the lymph nodes, we can capture and engulf pathogens and cellular debris and destroy them and prevent them from spreading throughout the body. Now, in addition to filtration of lymphatic fluid, our lymphatic system is also responsible for the filtration of foreign materials in the blood. So this still falls under point number 4, but rather than looking at lymph nodes, which filter lymphatic fluid, we're now looking at the spleen, which is responsible for filtering blood. And we'll see how this works later on in the chapter. Finally, point number 5, we have the production of lymphocytes, and we house our lymphocytes. And so our lymphatic system does both of those. We have primary organs of our lymphatic system, specifically our red bone marrow and the thymus, which are the site of production and maturation of our lymphocytes. So our red bone marrow is going to be responsible for the production of B cells and T cells, as well as the maturation of B cells. And then we rely on the primary organ called the thymus to mature our T cells. And then we'll look at our secondary lymphatic organs, which provide housing for these cells involved in an immune response. And we'll focus on this much more heavily next chapter with chapter 21. As we move through the remainder of this chapter's lecture, we're going to study the 5 main components of our lymphatic system. We're going to look at lymphatic fluid. We'll consider the various vessels that carry that fluid. We're going to consider the cells that are found in the lymphatic tissue. And then we'll look at the lymphatic tissues and finally the organs of the lymphatic system. We begin first with the lymphatic fluid or lymph fluid, or sometimes we'll just call it lymph. Lymph fluid is a clear or slightly yellowish fluid that's derived from blood plasma. And with this in mind, we're going to find water, which serves as a solvent for other substances. We're going to see a few plasma proteins, similar to what's found in blood plasma, but in very, very low concentrations. And it's worth mentioning here that some of those proteins may be coagulation factors and enzymes found naturally in the blood. We'll look at electrolytes, such as sodium, and in very low concentrations, potassium and calcium, which help maintain fluid balance and support cells. We might find lipids, antibodies produced by plasma or B cells. We might find amino acids, sugars, metabolic wastes like urea, or other waste products transported from the tissues to ultimately be excreted by the kidneys. Further, our lymph fluid contains macrophages and lymphocytes, as well as B cells, T cells, and maybe even dendritic cells. We might also find pathogens like bacteria and viruses which have entered the lymphatic fluid as a result of infected tissues. We might see cell debris, again from damaged tissue and damaged cells. Next, we can discuss the pathway by which lymphatic fluid takes in order to be filtered and return to circulation. Just as blood capillaries converge to form venules and then veins, what we're going to see is the lymphatic system. That transport system is designed as a branching system as well. And in fact, we're going to find that there are a handful of components making up our lymphatic transport system. We have capillaries. We'll then rely on the convergence of capillaries to form collecting vessels. Then we'll consider trunks before those trunks converge to ducts, which then dump fluid back into circulation. So let's go ahead and look at the components of our lymphatic transport system by considering lymphatic capillaries first. And so when we talk about lymphatic capillaries, we actually have two types of capillaries. We have our traditional lymphatic capillaries that are very intimately associated with blood capillary beds, as well as we have the lacteals, which are found in the microvilli of the small intestine and are responsible for absorbing dietary lipids and lipid-soluble vitamins. Compared to blood capillaries, lymphatic capillaries are slightly larger. They have a unique structure such that interstitial fluid may naturally flow in, but not out. And again, as I mentioned a moment ago, there are two types of lymphatic capillaries. So here we're going to consider our traditional lymphatic capillaries. So here's a capillary bed. We're going to see our arterial end here and our venous end here. So blood is moving in this direction. We'll push fluids out into the interstitial space as well as nutrients, gases, hormones, etc. And then we're going to pull a substantial amount of those things back into circulation here. But I said earlier, we have about 20 liters of fluid with which we push out per day. If we factor in all of our capillary beds, 17 to 18 liters pulled back at the venous end. What's left behind is pulled in courtesy of these green structures here that we call our lymphatic capillaries. Our lymphatic capillaries, if we zoom in here, there's the details of our lymphatic capillaries. We see it's a very well-designed system by which fluids can be pulled in, and when pressure is greater within this interstitial space than within the lymphatic capillary itself, the cells that make up this lymphatic capillary, so we have individual cell here and here and here, they're going to separate slightly, allowing interstitial fluid to flow into this lymphatic capillary. But when the pressure is greater within the lymphatic capillary, now we have a lot of fluid here and less fluid out here, this is going to put extra pressure on the cells making up our lymphatic capillaries such that the lymph fluid can't escape. And this is a very well-designed system to ensure that fluid flows in but can't flow back out. So we have, if interstitial fluid pressure is greater than lymphatic fluid pressure, then the endothelial cells are going to separate and interstitial fluid flows in. Whereas if the lymph fluid pressure is greater than that of our surrounding interstitial fluid, then endothelial cells adhere or tightly close and lymph can't escape via the interstitial space. Now, the other type of capillary bed I mentioned earlier, we have the lacteals. So these are specialized lymphatic capillaries located in the microvilli of the small intestine, which we haven't talked about yet, but if you imagine the small intestine is lined with these tiny projections called villi, which increase surface area, and the villi have little projections called microvilli. So we are going to see here an increase in surface area courtesy of villi and microvilli, and within those, we're going to see this green structure again. In our textbook, we represent green with a lymphatic system. And so we have blood capillaries that will reabsorb nutrients, and then we have our lymphatics, courtesy of these lacteals, which are going to be responsible for the absorption and transportation of dietary fats and fat-soluble vitamins from the gastrointestinal tract. Lymphatic capillaries are going to join together to create what we call collecting vessels, sometimes simply called lymphatic vessels. And these vessels contain valves, they direct fluid toward the heart, they pass through specialized structures called lymph nodes. We're going to examine lymph nodes later on in lecture, but at this point, recognize that lymph fluid is going to pass through nodes found periodically along these collecting vessels. And this will be the site of filtration of our lymphatic fluid. So we've gone from lymphatic capillaries, we've seen the merging of various lymphatic capillaries, now we're at the vessel level. And in association with that vessel level, we see lymph nodes, and we'll talk about lymph nodes in a little bit. As we progress and continue moving toward the heart, we're going to move from the lymphatic capillaries to collecting vessels. Now we've filtered lymph fluid via our lymph nodes, and ultimately our collecting vessels are going to converge or unite to form lymphatic trunks or lymph trunks, which serve as a major conduit that will drain lymph from various regions of the body. And we have 5 primary trunks in our body responsible for draining specific regions of the body. And so first of all, we're going to have this region here that's called the intestinal trunk, which is going to drain lymph from the stomach, the intestines, the spleen, the pancreas, and some of the liver. And the fluid associated with this region is often milky in nature because it carries a large amount of absorbed fats from the digestive tract. Next, we're going to see the lumbar trunk. And so as we consider the lumbar trunk, that's going to drain fluid away from our lower limbs, from the walls of the viscera of the pelvis, from the kidneys, from the adrenal glands. And so we're continuing to move up. So when we consider our intestinal and lumbar region, we are truly considering similar areas here. We're just draining different parts of the body. Lumbar trunk drains lymph from the lower limbs, again, as well as the walls and the viscera of our pelvis, from the region of adrenal glands and the kidneys, and from some of the abdominal region. And then we're going to have the bronchomediastinal trunk. So we're moving up superiorly, and this is a paired trunk that's responsible for draining lymphatic fluid from the thoracic region. So we're positioning ourselves here. So again, we're moving things superiorly. We also have the subclavian trunk, which is again a paired trunk here and here that's responsible for draining fluid from the upper limbs, from the shoulders, and some of the chest wall. And then finally, we have our jugular trunk, which is going to move inferiorly. It's a paired trunk responsible for draining fluid from the region of our head and our neck. And the goal is to dump fluid here and here. And I'll show you that in our next slide. Again, I've shown you those 5 trunks. Now we are moving to the ducts themselves. Now, the ducts, if we consider the ducts, lymphatic fluid is ultimately delivered to circulation via 2 very large lymphatic ducts within the thoracic cavity near the heart. We have the thoracic duct, which is sometimes called the left lymphatic duct. So here we're looking at our subclavian vein, right and left. When we talk about our thoracic duct, sometimes called that left lymphatic duct, here we're dumping lymphatic fluid into our left subclavian vein. And then here we have our right lymphatic duct dumping lymphatic fluid into our right subclavian vein. Ultimately, the fluid being dumped by these 2 ducts is dependent on the origin of the fluid, and we'll find that it's very asymmetric in nature. When we consider our left lymphatic duct, sometimes referred to as the thoracic duct, this area or region in yellow, that duct is going to receive lymph from the left side of the body. So we have the head, the neck, the chest or the thoracic region, upper left limb, as well as both lower limbs, draining fluid. And in contrast, if we look at our right lymphatic duct, we're going to see that it's going to drain lymph from the upper right side of the body only, which includes the head and neck of the right side, our right arm or upper limb, as well as the thoracic region, right side only. So fluid from this region is going to be dumped via the right lymphatic duct. We've looked at the part of the lymphatic system that carries lymphatic fluid. Now I want to go ahead and look at the cells of our lymphatic tissues, where we're going to find 4 major cell types. First, we have lymphocytes. Remember from last term, lymphocytes are one of our white blood cells that we studied back in chapter 17 in our study of the blood. And lymphocytes are part of our adaptive immune response. That is, it's not something we're born with, but something that develops as we're exposed to pathogens. So yes, we're born with lymphocytes, but not specific to a given antigen or pathogen. And we'll learn more about the adaptive immune response in next week's lecture. For now, know that lymphocytes function by providing an immune response that either neutralizes or directs or mounts some kind of attack against a specific foreign substance that we call an antigen. And there are 2 major classes of lymphocytes. We talked about this last term as well. We have B cells and we have T cells. So first, our B cells, or our B lymphocytes. B cells are going to originate and mature in the red bone marrow, and they have 2 primary functions. First, they produce, or I should say they secrete, antibodies or immunoglobulins, which help neutralize foreign threats in the body, often discovered and presented by specialized cells like macrophages or neutrophils. And second function, they serve as antigen-presenting cells for a T cell to respond to. So ultimately what they do, B cells are going to show a T cell what they found that might be harmful. Maybe it isn't, maybe it is, but they show what they found to a T cell. And B cells are involved in what we call the humoral immune response, a branch of our adaptive immune response we'll study next chapters. There's actually a whole branch of our immune system that's dedicated to B cells. We have 2 types of B cells. We're going to see plasma cells, which go on to secrete antibodies designed for very particular threats. And then we're going to have memory cells, which help us remember a prior foreign threat. So something we've seen before, it will remember what we've seen so that we can mount a response to that same foreign threat more quickly, more swiftly, as compared to the first time we discover it in our body. We also have T cells originating from our bone marrow, just like our B cells, but now we're going to mature in the thymus. T cells are responsible for what we call a cell-mediated immune response, the other branch of our adaptive immune response we'll see next chapter. And T cells do a number of things. First of all, we have helper T cells, which direct other cells to do something. They often give permission for other cells to do their job. And we have regulatory T cells, and not to confuse this with helper T cells, but they're going to not give permission, but they're going to suppress things. They're going to suppress an immune response, which is especially important in ensuring we don't develop some kind of an autoimmune disorder or otherwise attack self cells. So regulatory T cells are going to suppress something. Helper T cells are going to give permission for something to happen. We have cytotoxic T cells, and if their name doesn't give it away, well, they're just cytotoxic. They detect virally infected cells, they detect cancer cells, and they kill them through a process of controlled cell death that we call apoptosis. And finally, we have T cells called memory cells. So we saw this with B cells as well, and these memory T cells are going to help us remember prior antigens we've been exposed to before, allowing us to mount a response to something foreign much more quickly, much more swiftly, much more aggressively as compared to the first time we see something in our body. And there will be more discussion on T cells and memory T cells specifically next chapter. The second cell type found associated with the lymphatic system is called the macrophage. Macrophages are distributed throughout our lymphoid organs and the connective tissues of our body, and they begin their life as a monocyte, one of our white blood cells in the blood, but then they leave the bloodstream to begin their life as what we call a macrophage in some kind of tissue, responsible for looking out for foreign materials or cleaning up cell debris. Macrophages serve as phagocytes. They engulf pathogens, they engulf cell debris, they engulf dying host cells, they digest them, and then they drag what they found along to the lymphatic system. And specifically, they present what they found to B cells and T cells in the lymph nodes, presenting parts of what they phagocytized. So they just present little pieces of what they have found. Macrophages are one of the first responders to tissue damage, and they make up what we call our innate or non-specific immune response, something we'll learn about next chapter. Macrophages, in addition to their role as phagocytes, are also involved in antigen presentation. And in fact, after they phagocytize a pathogen, they take little fragments of the pathogen, present these fragments on their cell surface, and that's what they show to the B cells or T cells to let those particular cells, the lymphocytes, know what they found. And if conditions are right, our B cells or our T cells are going to respond. Now, again, we'll learn about that next chapter. So for now, just recognize that macrophages are phagocytes and macrophages are antigen-presenting cells, and they begin as monocytes in the blood. Our third cell type of the lymphatic system is the dendritic cell. We've seen dendritic cells once before when we were looking at chapter 5 of the integumentary system back in the first term of anatomy and physiology. Dendritic cells are found throughout our tissues where they're involved in the normal tasting and sampling process of their surroundings. They tend to have contact with tissues that are connected to the outside environment, like our skin, the dermis and epidermis, as well as the mucous membranes that line our respiratory tract, our gastrointestinal tract, the urinary tract, and some parts of the reproductive tract. As dendritic cells come across some kind of antigen, like a bacterium or bacterial toxins, maybe pollen, dust, a virus, they phagocytize the foreign material, and then they migrate out of the tissues and enter the lymphatic system, where, just like macrophages, they drag what they found to our lymph nodes and present what they found to the B cells and T cells, those B and T lymphocytes that are found there. Finally, we have reticular cells. Now, reticular cells aren't immune cells as we saw with our lymphocytes, macrophages, and dendritic cells, but rather they contribute to the structural and functional integrity of our lymphatic tissues by creating a soft internal skeleton made up of a fine matrix or network of reticular fibers composed of collagen. The framework serves as a scaffold to house immune cells. It helps route fluid, be it lymphatic fluid or in the lymph nodes or blood in the spleen. And so we're seeing a routing through our lymphatic tissue. Those reticular cells create that scaffolding to serve as a filtration system to help trap pathogens and particulates such that phagocytic cells can remove them. And so this should be a reminder back from the first term of anatomy and physiology, where we looked at reticular fibers as well as the tissues that make up the spleen and our lymph nodes. We move now to the study of lymphoid tissues. These tissues, which are part of the immune system, come in two forms. We have what's called diffuse lymphoid tissue, and then we have lymphoid follicles, sometimes called lymphoid nodules. Our diffuse lymphatic tissue is found throughout the body, but it's especially dispersed along our mucous membranes of the gastrointestinal and respiratory tracts. These tissues consist of reticular fibers made up of a very loose framework, diffusely infiltrated by lymphocytes and macrophages, and this tissue provides a point of surveillance for the immune system, standing guard and ready to induce an immune response as an antigen penetrates the tissue. In contrast, we have lymphoid follicles or nodules composed of the same reticular framework and containing the same kinds of cells as our diffuse lymphatic tissue. However, the lymphocytes are more numerous, more closely packed together. They house special regions called germinal centers, which we're going to cover next chapter. And further, they're surrounded by capsules, giving them very distinct boundaries from their surroundings. So here we're going to see our follicles, and I've highlighted this. This slide comes from the A&PIII site, where we're going to see very distinct regions. So this is a follicle, and then we have more diffuse tissue here as we're looking at some of the tissue of the appendix. We've looked at lymphoid cells and lymphoid tissues. Now let's go ahead and step forward in our study of lymphoid organs. Our lymphoid organs are divided functionally into primary and secondary lymphoid organs, each serving a very distinct role in the development, maturation, and function of lymphocytes. And our primary lymphoid organs are sites where stem cells divide. We say they go on to produce mature lymphocytes. The primary lymphatic organs include the red bone marrow and our thymus. So first of all, red bone marrow. Stem cells in our red bone marrow give rise to mature B lymphocytes as well as to undifferentiated, immature T lymphocytes. We call these T cell precursors. And the T lymphocytes then migrate to and become mature in the thymus. And so that brings us to the thymus. And if we're looking at the thymus here, so here's our thoracic cavity, lungs, heart, we're going to see our thymus positioned here. The thymus is the site of differentiation for T cell precursors where they become mature. And so I want to be clear here. We see both our B and T lymphocytes originate in the red bone marrow. Our B cells go along to mature there as well, whereas our T cells must travel to the thymus to undergo maturation. And curiously, the thymus gland is relatively large at birth. It continues to play a role in early development of the body's immune system. However, following childhood and early adolescence, the thymus begins to shrink, degenerating further into old age. And despite its atrophy, the thymus does continue to provide immune support throughout life, although at a declining rate as we age. In contrast to our primary lymphoid organs, where we see the development and maturation of B cells and T cells, we also have secondary lymphoid organs. And so I've listed those secondary lymphoid organs here. Secondary lymphoid organs are sites where most immune responses occur. And in our study this week, we'll look at three major secondary lymph organs: our lymph nodes, the spleen, and tissue we call MALT, or mucosa-associated lymphoid tissue. That includes our tonsils, the Peyer's patches of our small intestine, and our appendix. As we focus our attention on the secondary lymphoid organs, I want to look at lymph nodes first, and we'll spend most of our time considering lymph nodes. Lymph nodes are an important secondary lymphoid organ where they function to filter lymphatic fluid, ensuring fluid is clear of pathogens and cell debris prior to returning it to the bloodstream. Second, as lymph fluid is filtered by the lymph nodes serve a role in the body's immune response where fluid passes by resident macrophages that are housed in the lymph nodes, such that pathogens and cell debris found in the lymph fluid can be removed through a process called phagocytosis. So first two functions, we filter the fluid, and we also see the lymph nodes house macrophages, which are responsible for filtration. Finally, lymph nodes serve as a site of activation for our B cells and T cells. Specifically, a mature B cell or T cell, once they encounter their one unique and particular antigen, can become activated. And it takes a bunch of signals for that to happen beyond what's discussed in the scope of this course. But this is the site of B cell or T cell activation, which leads to the proliferation of an army of B cells or T cells and the subsequent differentiation of those B cells or T cells in order to mount some kind of an immune response. And here is an image of a lymph node. We're going to look at the structure and the fluid pathway in a few slides from now. We say lymph nodes are bean-shaped structures found scattered throughout the body along lymphatic vessels, like beads on a string or pearls on a string. And there are more than 600 of these nodes scattered throughout the body. Yet, despite being scattered, they're certainly found with more density as clusters in 5 major sites of the body. First of all, we're going to see the cervical region here at the neck. We'll see the axillary region, the underarms. We're going to see a mesenteric region associated with the abdomen, a vertebral region associated with the thorax here, and then we're going to see an inguinal region associated with the groin. And some of our lymph nodes are described as being superficial, that is located near the surface of the body, such as at the neck, underarm, and groin. And these are the lymph nodes we can palpate. Sometimes described as swollen glands, enlarged lymph nodes may indicate an immune response, an infection, or even some form of cancer. Lymph nodes are designed to maximize filtration of lymphatic fluid and help facilitate an immune response. First, lymph nodes are surrounded by fibrous connective tissue in the form of a capsule. So we will see the capsule here, and we're going to see projections of tissue we call trabeculae, individually a trabecula, but we'll call them plural trabeculae, extending inwardly from the capsule toward the center of the lymph node that's going to divide the node into various compartments. Internally, the lymph node is divided into 2 major regions. We have an outer cortex, we have an inner medulla, and some textbooks might describe the cortex in 2 parts, an outer cortex and an inner cortex. For this class, we'll simply look at the cortex, or that outer region as a whole. The cortex contains lymphocytes in dense clusters. So we're going to see these dense clusters here within the cortex at these lymph nodules or lymphoid follicles. And in these nodules, we have B cell rich regions where the function of these B cells is to produce antibodies, which ultimately tag pathogens for the destruction. We also have T cells in the cortex, the main site for T cell activation and interaction with antigen presenting cells, which we'll talk about more later on next week. But just for now, know that antigen presenting cells have been macrophages, dendritic cells, that have picked things up in the periphery and drug them back into lymphatic fluid to be presented here at the lymph nodes. If we look at the medullary region or the medulla here, this region contains macrophages which spend their time on what we call medullary cords or inward extensions from the cortex into the medulla. So we have these dense clusters of lymphocytes, plasma cells, which are B cells that can produce antibodies, and of macrophages. So we have these medullary cords. And now that we understand the structure of the lymph node, I want to go ahead and talk about the pathway lymph fluid takes as it passes through lymphatic vessels from node to node. And as we consider that, we would see fluid traveling in this direction, moving and arriving into a lymph node via afferent lymphatic vessels. So afferent, so an arriving vessel. And then we'll see fluid penetrate the convex side of our lymph node here. Usually there are a few points of entry. So here we have 1, 2, 3, 4, 5, 6 afferent vessels by which fluid moves into the lymph node. Next, fluid is going to move through a series of sinuses. First, we're going to see right underneath the cortex, we have the subcapsular space or subcapsular sinus that's found just immediately below the capsule. And then we're going to see lymph flow through these trabecular sinuses. So we'll see fluid moving in this direction. That's going to extend through the cortex parallel to the trabeculae. Finally, lymph enters the medullary sinuses, which extend through the medulla and provide a route for fluid. Lymphatic fluid ultimately is going to drain back out of our lymph nodes via typically one individual efferent lymphatic vessel and leaves the lymph node by way of this region here called the hilum. So our efferent vessel is found at the hilum. And then we're going to continue the path along more lymphatic vessels, arriving to more lymphatic nodes as this fluid makes its way back to circulation. Taking a step back, let's go ahead and review what we've learned up through now by looking at the bigger picture of lymph fluid formation, collection, and routing back to circulation. In doing so, we trace the path of fluid from blood capillaries to the subclavian veins. And we begin by considering blood in our capillaries. So we'll look at blood in our capillaries, at which point, with the help of blood pressure, blood plasma and some solutes, nutrients, oxygen, they're going to be filtered out of our capillaries and into the interstitial space. And so we've now formed interstitial fluid. So we began with blood in our blood capillaries. We push some of that blood out into the interstitial space, creating interstitial fluid. Thereafter, our lymphatic capillaries are going to pick up that fluid and any plasma proteins that might be in the area to create lymph fluid. Lymphatic capillaries are going to merge to those lymphatic collecting vessels or lymphatic vessels, as the lymph fluid must be filtered as it's routed back to circulation. So we're going to see movement through lymph nodes. Beginning the filtration process, lymphatic vessels route fluid along to those lymph nodes courtesy of afferent lymphatic vessels. At the lymph nodes, we're going to still see lymphatic fluid. We see that fluid leave via efferent lymphatic vessels. Again, we're still lymphatic fluid. Then those vessels are going to merge to create trunks. We still have lymphatic fluid. At that point, we're going to see our left and right ducts, so our thoracic and right ducts, still lymphatic fluid, at which time we're going to dump that lymphatic fluid into circulation via the subclavian veins. And upon dumping lymphatic fluid into the ducts, fluid is returned back to circulation. So you should understand this pathway where we begin with blood, interstitial fluid, then we have lymphatic fluid being routed before dumping that fluid back into and seeing blood once more. As we look at lymphatic vessels, we'll find that they share many similarities to blood vessels in our study of chapter 19 of our textbook. And in fact, lymph vessels carry fluid. They're lined with endothelial cells. They rely on contraction of smooth muscle to aid in the movement of fluid, and they further rely on pressure changes and skeletal muscle contractions tied to breathing to help fluid move along. So you see many similarities between our lymphatic vessels and our blood vessels. Lymphatic vessels are distributed throughout the body just as our blood vessels are. They aid in immune function, not strictly by carrying immune cells such as we saw with blood vessels, which they can and they do, but because they carry lymphatic fluid to lymph nodes, pathogens can be filtered and the adaptive immune response can be initiated. So with these similarities, I want to point out one of the major differences between the two vessel systems, that of not having a pumping system. That is, we don't have the heart to rely on as vessels do. Rather, the movement of lymphatic fluid from the interstitial space and along to the upper thoracic cavity is accomplished through a series of systems, most of which are familiar to us as we recall low pressure veins of the circulatory system and the efforts needed to get that low pressure fluid back to the heart. With that in mind, we have 5 ways by which we move lymphatic fluid here through lymphatic vessels. First of all, we look here at milking. This is just a revisit of the muscular pump from the unit on the cardiovascular system, where contraction of skeletal muscles during body movement compresses lymphatic vessels, pushing or forcing fluid through the lymphatic vessels, often described as a milking action. And this is especially important for fluid transport in tissues below the subclavian veins. Next, we have one-way valves that line the lymphatic vessels, and like those found in veins, lymphatic vessels contain valves to prevent the backflow of lymph fluid. When lymph fluid is pushed forward as a result of milking or other forces, the valves close to ensure that fluid only moves in one direction toward the ultimate destination of the heart. So similar to what we saw with veins, we see these little cusps creating valves. Also, we're going to rely on pressure changes here. This is also a revisit from the unit on the cardiovascular system, where pressure changes in the thoracic cavity caused by breathing, that is, from inhaling and exhaling, aid in lymphatic fluid flow. Specifically, during inhalation, when we inhale, we're going to see the volume of our thoracic cavity is going to increase, pressure will decrease, and a vacuum effect is created where lymphatic fluid is drawn up by or otherwise flows from our abdominal region here. Also, it helps that as we inhale and increase the volume here, we're going to decrease volume of our abdominal contents. That is going to increase pressure and push fluid moving upward. Afterward, when pressure changes during exhalation and the abdominal region experiences a drop in pressure and an increase in volume, the valves of the lymph vessels prevent backflow of lymph. Further, we can also rely on the contraction of smooth muscle found in the walls of some of our larger lymphatic vessels, which contract rhythmically in peristaltic movement, propelling lymph fluid forward. And then finally, we have pulsation of nearby arteries. So we're looking here at our abdominal aorta, and we would consider the expansion and the contraction of these major arteries due to pressure waves generated with each heartbeat as a way to move fluid or lymph fluid along, specifically in the trunk of the body. And in some cases, some of the biggest parts of our limbs, where there's a close anatomical relationship between lymph vessels and arteries. In fact, we see lymphatic vessels running alongside arteries such that arterial pulsation compresses lymphatic vessels, aiding in the movement of lymph fluid. Now, none of these activities alone may be sufficient to move large volumes of lymph fluid, but when working together, these activities can contribute significantly toward the overall propulsion of lymph fluid toward the heart, despite the lymphatic system not having a pumping system of its own. We turn away from lymph nodes and consider the spleen, described as the largest single mass of lymphatic tissue in the body, and located on the left side of the abdominal cavity, directly beneath the diaphragm and connected to the stomach. We could describe it more anatomically as being found in the left hypochondriac region. The function of the spleen is three-part. First, the spleen acts as a blood filter, where it removes worn-out red blood cells. We've learned that already. Remember, red blood cells have a circulation of about 90 days, and as red blood cells become less flexible and less able to twist up so as to move through our small capillary beds, they're caught in the spleen, where they're broken down with the help of macrophages, and some of their parts are recycled. Further, the spleen acts to remove bloodborne pathogens from the blood. In the next slide, I'll show you that we have resident macrophages that spend their time filtering blood as blood moves through the spleen. And so those macrophages are going to help detect and remove foreign pathogens like bacteria and viruses from the blood. The spleen also serves as a reservoir, and as such, it becomes a reservoir for blood plasma, where it holds a significant volume of blood that can be released into circulation with any kind of sudden or significant blood loss, or maybe during exercise. Such a reserve can help maintain blood pressure with blood loss, as well as help ensure oxygenated blood reaches vital organs under conditions of hemorrhage or during intense exercise. And because it's a site for storage of blood plasma, the spleen also serves as a storage site for platelets, such that if there's ever a sudden loss of blood, the spleen contracts, blood is pushed out into circulation, and about 25% of the total platelets in our body are stored in the spleen. So with the release of this blood, we can better ensure that if we need to form blood clots, we have the ability to release platelets to do so. And further, as a site of storage of blood plasma, we also see the spleen as a storage site for red blood cells, which help provide for homeostasis in the body, specifically when the body activities demand more oxygen. And then finally, we see the spleen act as a housing site, so an active housing site for our immune cells, specifically macrophages and our B and T cells. And with this in mind, we return back to the point I made up here. Macrophages and lymphocytes help filter blood as it passes through the spleen. Lastly, I do want to point out the spleen serves as the production for red blood cells during early fetal development before we have red bone marrow to rely upon. And we touched upon that in the very first term of anatomy and physiology when we were studying bone. Now that we can recognize the general functions of the spleen, let's go ahead and look at the structure of the spleen. And to do so, I'll start by saying that the tissue structure of the spleen is similar to that of our lymph nodes. The spleen is surrounded by a capsule of dense connective tissue, which serves or helps to protect and maintain the shape of the spleen. And superficial to this, it's covered by visceral peritoneum, which allows the spleen to be more slippery as it interacts with other organs in the same vicinity, including the left kidney, part of the colon, and the stomach. And the spleen has a hilum. So we're looking here at the hilum, this concave area found here, which serves as the entry and exit point for the splenic artery and the splenic vein, as well as lymphatics and nervous tissue. We also have red pulp and white pulp. When we consider pulp here, our white pulp, if we see in this kind of late orangish pink color here, or this lighter color, this lighter region here, white pulp is lymphatic tissue surrounding some of the arteries within the spleen, and it houses resident immune cells like our macrophages, as well as our lymphocytes, the B and T cells. Functionally, the white pulp provides a site for lymphocyte proliferation and for immune surveillance. And so with this in mind, we see lymphoid follicles or nodules, similar to what we saw with lymph nodes, that contain B cells that develop germinal centers where B cells can go on to proliferate and produce antibodies in response to being matched to their one unique antigen. And in contrast, we also have red pulp. So this is the darker region here. Here we have the darker, more purplish region here. We're going to see that this red pulp consists of sinusoids, which are blood-filled spaces where blood is filtered, and it kind of pools in this region, as well as it has a network of what are called splenic cords, which help not only in the storage of blood, so that's going to help us with blood plasma, platelets, and red blood cells we mentioned already, but also it houses macrophages needed to filter out and remove old, worn out red blood cells, degrading them, prepping them for recycling, as well as removing and filtering out pathogens and debris found in the blood. We can consider blood as it moves through the spleen. So here we're going to see a zooming in of the spleen, by which we're going to see blood arriving to the spleen via the splenic artery and ultimately exiting out the splenic vein, both of which are found arriving and departing from the hilum of the spleen. So we can trace our splenic artery. So we're going to trace it here. We're going to see it's going to divide into smaller branches called trabecular arteries, which run along trabeculae. These are going to be partitions that extend from the spleen's outer capsule within the interior of the spleen. So we saw similar structures when we looked at our lymph nodes earlier in lecture. And our trabecular arteries then are going to further branch into central arteries, which are surrounded by lymphoid tissue. So we're going to see white pulp in this region here. And then from here, our central artery branches into arterioles, which then finally drain into the sinusoids, which ensure that blood passes through very slowly, allowing macrophages to remove old or otherwise damaged red blood cells and pathogens and debris from the blood. Blood is then going to collect within these collecting sinuses, or sometimes you might see them called collecting veins within the red pulp, which then aid in drainage of blood from the spleen out via trabecular veins, which run along with the trabeculae again, sending blood toward the hilum along the splenic vein and out of the spleen. And so overall, this sluggish vascular design provides for a filtering system which allows for the interaction between blood and cells of the immune system. And as blood flows through the various spaces, it comes into contact with the cells of our lymphatic tissue, including our macrophages and our B cells, our T cells, that help initiate the immune response. We have just a few cells left where we're going to talk about secondary organs called MALT, or mucosa-associated lymphoid tissue. These are found along various mucosal lined surfaces, and they serve as part of a first line of defense against ingested or inhaled pathogens. We find these tissues associated with the digestive mucosa and respiratory mucosa, as well as we find these tissues associated with our urogenital mucosa. And some MALT tissues are simply small, solitary nodules positioned somewhat diffusely, scattered within the mucosal lining, whereas other malt tissues are found in larger aggregates in specific places throughout the body. And these more organized or defined malt structures are some that you might know are tonsils. You may have heard of Peyer's patches or Peyer's patches, and the appendix. So first we have our tonsils. These are partially encapsulated aggregate tissue. And we have a group of tonsils ultimately forming a ring of lymphoid tissue around the pharynx. And this ring includes the pharyngeal tonsils, our palatine, and our lingual tonsils. So we'll see our pharyngeal tissues, we'll see our palatine at the very back of the soft palate, and our lingual tonsils near the back of our tongue. We also have Peyer's patches, aggregates of unencapsulated lymphoid tissue found embedded in the walls of the ileum of the small intestine. And Peyer's patches form an important part of our immune system by monitoring intestinal bacterial populations and preventing growth of pathogenic bacteria within the intestines. And lastly, we have the appendix, formerly known as the vermiform appendix. It's a small pouch-like sac of tissue located in the lower right quadrant of the abdomen, positioned close to the junction of the large intestine and small intestine. And the appendix contains dense lymphoid follicles or nodes, rich in immune cells like B cells and T cells that are thought to play a role in monitoring and responding to pathogens within the gastrointestinal tract. Further, the appendix is thought to act as a reservoir for the flora of the digestive tract, repopulating the digestive tract when needed in order to maintain a healthy microbiome. And I'd really like you to focus on those two functions as compared to some of the material that our textbook outlines. So as we close the lecture for this chapter, I want you to take a moment to consider the strategic positions of our secondary lymphoid organs. They're positioned throughout the body so as to intercept invaders that enter the body via different routes. Think about it. If you develop a bloodborne infection, the spleen is going to help filter out a bloodborne pathogen. And in contrast, if a pathogen breaches the skin or a mucous membrane, antigen-presenting cells deliver antigen to our lymph nodes. If you inhale or ingest a pathogen, your tonsils might help remove the pathogen, or the malt of the respiratory or gastrointestinal tract can generate an immune response. So position is everything in terms of the secondary lymphoid organs. And with that in mind, we're going to conclude our lecture of Chapter 20. When we return next week, we're going to discuss the immune system, where we'll rely heavily on what we've learned this week to broach the topics of the immune system. If you have any questions, please don't hesitate to reach out to me during office hours. And meanwhile, make it a great day.